Okay, so this morning's talk is about segmentation. The PDF of the slides is on your GitHub already. I think some of you have taken a little look. But segmentation is non-trivial, and it's probably the most important part of a image analysis pipeline. If you get the segmentation right and you do a good job of it, then the rest of the analysis is, generally speaking, quite straightforward. And if you have a good segmentation, then your interpretation of the data can often be less ambiguous. So it's an important part to get right from the beginning. Here we have an image, and this is what you may imagine a kind of typical segmentation pipeline to look like. So we have a grayscale input image, and then we do some kind of thresholding, a segmentation to separate into foreground and background. And then we do some kind of labeling. So these background and foreground regions are labeled on the instance level. So we call this a semantic instance level. The semantic is the background and the foreground, and then the instance level is turning the cells into individuals so that we can take independent measurements from each one. But don't take this to be the only form of segmentation. This is one aspect of segmentation, and we generally call this, if we're doing histogram analysis to it, we generally call it thresholding. But segmentation is bigger than this notion of going from grayscale to a binary interpretation of our image and then our labeled cells. And I'll show you some other examples. So, for example, this would also be included in segmentation. What we're doing here is we're segmenting out the maxima. We're finding the points, the bright points, the punctate foci in these images. This is also segmentation. Also, what my research focuses on a lot at the moment, which is learning bounding boxes for cells. This is also segmentation, object detection. And then we have things like finding lines in an image or circles in an image, shape detection. So this is also segmentation. And finally here we've got region detection, where we try and group regions based on their visual or spatial similarity. Okay, so we're going to cover all of these in more detail, but don't think of segmentation as just this turning your image into a binary representation. It's a bigger subject. So this is a sort of pipeline which you'll have for all segmentations. So we start with our image import, which we covered a lot yesterday. We're all using this SK image library, and we're using the import-output functionality of that to get our image in. And what we're essentially doing is that we are reading the numbers into our Python environment so that we can start processing them in different ways. And then once we've got our image in, then it's often necessary to do some kind of formatting to the data, some kind of pre-processing. So we may split the image into different channels. So an image that appears blue may well be a free channel image, so you just want to extract the meaningful channel from that. Or you may have an image with multiple channels with information included, and we still want to selectively take channels from that. So generally there's some kind of splitting that goes on when you import a raw format from a microscope. And then again, you may decide that you want a 32-bit or 64-bit format for your image, so you'll convert it into a floating point format so that when you do processing, you don't lose precision when you start doing divisions and multiplications and things like that. The floating point is a safe format for when you're doing mathematical operations on an image. The constraint is, as you heard, that it takes more memory, and it could, in theory, be slower if you were doing tens of thousands of images. But if you want to be careful, then it's routine to have a step where you convert generally from an 8-bit or a 16-bit into a floating point format like 32 or 64-bit. And then we have our segmentation. So this can vary, as I've told you, but essentially there'll be some kind of algorithmic process which will convert our image from one format and transform it into another space, and may also have some sort of post-processing which helps split the cells or the objects that you're looking at. So this is really the subject of this lecture. But once you've done some segmentation, then it's often necessary to do some kind of labeling where we split our semantic segmentation into these regions, and then we do we're able to isolate these and index them so that we can refer to them in our subsequent analysis, which in this case is almost invariably taking measurements from our image. So it's generally typical to go back to your raw data and to use your thresholded segmented image as a means of isolating regions and taking measurements from there. So we may be measuring the mean intensity of these regions or the maximum intensity. If we're measuring the area, then we can do it from this image, but there are a lot of measurements you can do on the raw image and also subsequently on the thresholded image, but there's generally some necessity to go back to that earlier version of the image, which is more close to the raw measurement. So you can see this as being like a universal pipeline, and during the practical you will go through this on a certain number of images. Okay. Feel free to ask questions as we go along. That's fine. So scikit-image is a great library for basically doing all of these steps, because you can program all of this in sort of raw NumPy or in original Python, but this has a lot of functions which make life a lot easier. So you don't really have to go into the image and identify, you don't have to write your own flood fill algorithm to convert these foreground and background regions into independent cells. There are functions in scikit-image which can take the edge off doing that stuff yourself, and people have written optimized code to do it. So although it's an interesting exercise to write a lot of that yourself, you can save yourself a lot of time if you're pushing a deadline or something and use this library. So this features a lot in the practical after this talk as well. So we'll start with simple numerical segmentation. So this is probably what you're most used to if you've done Fiji Image Day, and we talked about it briefly yesterday as well. It's called image thresholding. So say this is our original image. What we want to do is, first of all, interpret this, convert it into a histogram representation. So in a histogram, rather than looking at the pixels arranged spatially, what we're doing is we're counting the number of pixels of a certain intensity. So you see this distribution has two peaks. It has a peak here which represents the background pixels. So there are a lot of black pixels here. And then there's another peak here which represents the sort of most prominent fluorescence in the cells. Okay? And this forms the foreground. And what we want to do is we want to choose a threshold which allows us to separate the background from the foreground, like separate it into two sets. And this line here represents where we might make our decision to be. Okay? And what this will do, will create a binary version of the image where we basically labeled things as true or false, true representing foreground cells and background as being a 0. Now you might see this here. This represents a little bit of saturation in our image, and you might see that in the practical later. So always keep an eye on what's going on, but this is quite a small amount, so we're going to proceed with the analysis. Okay. Now, in the previous example, I just came up with a value and put a line there, but in reality, you'll typically use an algorithm which will interrogate your histogram, and it will suggest a threshold which is most appropriate for what it thinks the image is. Now, it would be great if we could always just choose a threshold ourselves and keep that value, but because anyone that's done immunohistochemistry or cytochemistry, they'll quickly realize that samples are very variable from day to day. Staining efficiency is different, and so you want a technique which is somewhat flexible. So ideally you would just choose one value and keep to that. That's very scientific, but invariably we'll use an algorithm which will dynamically interrogate our histogram, which is related to the quality of our staining, and it will give a reproducible answer for where it thinks the threshold should be. So it's not as good as a fixed value, but in terms of its scientific reproducibility, but it's reproducible because it's an algorithm, and it will always give you the same answer for a histogram. Okay, so what this allows is, it allows you still to be reproducible, but it also encourages the process to be somewhat dynamic, reacting to how good the image is. Obviously, if your image is extremely faint or extremely bright, then no algorithm will save you. But this just allows you to incorporate a bit of flexibility into your pipeline, which can allow you to accommodate some of the biological noise in your experiment. So we tend to use algorithms like this, which can interrogate the histogram and kind of give a reproducible answer no matter what it sees within limits. Okay? So there are some famous examples. There is, for example, the Lie algorithm, which is this one, the mean, ISO data, minimum, Otsu. Otsu is probably the most famous. And all these algorithms are quite simple. They are kind of looking at the means of the distribution, trying to separate them in an iterative sense, or they're looking at moments of the distributions. They're all very simple. Basically, choose an approach which is sensible and keep to it for your experiment for both conditions, and just be aware that you might be undermining your experiment if you choose something that's too specific to what's happening. But we'll talk more about that as we go along. So in the practical later, you'll see the Otsu algorithm. This is a very famous one. You can look up online how it works. It's very simple, but it seems to work very well. And here we've got a bit of a description of how it works. So for each possible threshold, we walk through, and then we calculate the variance in the background intensity. So all of the at that particular potential threshold, all the variance in the background pixels and in the foreground pixels. And then we weight each of these variances by the number of pixels that contributed to either foreground and background at that threshold. And then we retain a threshold which gives the smallest variance across all of the thresholds. So we kind of tune it to be the sweet spot between the number of pixels being included and the variance in the raw pixel values within there. And this seems to work remarkably well. So it's become quite famous. So that's as far as we're going to go with these numerical methods. They're sort of used very often, but they're not very powerful. They are dependent on just numerically interrogating the histogram of your image, and there's a lot more information in an image which can help you inform you for your segmentation. So this is like the first go-to step, and if this works, it's fantastic, but generally speaking, you have to go a bit more advanced. So when we do this, it's helpful if you start thinking about your image in a slightly different way. And a great way to think about your image is if it's a topographical 3D image. So what I mean by that is, so when you see an image, it goes from dark to light, and this is fine, and you can visually make sense of this. But what I want you to start doing, and this is kind of what I do, is I start to think of it as a kind of 3D surface where it's light, we have like a mound, and where it's dark, we have a kind of a basin, we have a lower area. So this is like a topographical representation. So you can imagine sort of contours on a map almost. And the brighter it is, the taller it is, and vice versa. So hopefully you can all see how the top row and the bottom row are connected in these images. And it's useful if you can start thinking in this way, because it's an idea that's used a lot in terms of segmentation. Now, although I say the bright areas are always the peaks, that's not actually the case. You can flip between the two. So sometimes the brightness represents the basin rather than a peak, and that just depends on the algorithm how they formulate it. But just see it as a different elevation compared to the other shade. Okay. So here we're going to talk about the Watershed algorithm, and here we actually consider the bright areas. We actually invert the image and consider the bright areas to be basins. Okay. So imagine this is two peaks, but we've just inverted it. And then what we do with the Watershed algorithm, when we use this algorithm for separating blobs that are very close to one another, what we imagine is that each basin, in this case, which would be the centers of our bright objects, we imagine we start filling them up with water based on how intense they are. So we start here in the case of the darkest areas, and we give each basin a unique color, and then what we do is we start filling up the basins until they touch. And this represents the closest point that these two cells are along their perimeter. And so you can imagine this is kind of the idea. And what this allows us to do is, using the brightest points as seeds, we can then sort of flood fill our image, taking into account how the gradients of the intensities change, like where the steep areas are, and it allows us to create borders between our cells. You might have used the watershed briefly yesterday, and you'll use it again today in the practical, and it's a great way of separating images where you thresholded them and you want to find the boundaries between these cells that actually touch. So we've got an example here. So this is actually a cell where we've done the thresholding, and then within the thresholded area we've then done watershed. And what we've done is we've picked as our basin centers the brightest spots, and we've filled up the cells incrementally, and we can see we get something like this. This is not very good, but you can see how it works. Even though the cells are linked, we've managed to sort of segment them. But because the fluorescence here is quite ununiform, this idea of hills and basins doesn't work quite so well, because this is like this isn't like an Everest style peak. This is like, you know, something in the Lake District, which is a bit kind of more undulating, and so it's not a clean thing. So what we tend to do is, rather than actually using the intensity levels, we actually use a different measure of where the centers of these objects may be. And a very common approach is actually to do the watershed on a distance transform of our image. So what we do is we segment it, and then what we do our own distance transform. So we start at the edge and we start counting up. And we go round and we count up, and then once we've got round to the beginning, we go up another increment and we keep going round until in the center of our objects we have these peaks. So this is a distance transform. And so what we do is we apply the watershed not to this intensity distribution, but to this distance transform. And then you can see we get a very nice separation of our objects, which is based on distance, not just intensity. Okay, so this might seem like an extra step, but it's so common to see watershed applied in this way. But actually, I want you to know that watershed can be applied to raw intensities as well. But this is commonly what you end up doing. So the watershed is the process of incrementally flood filling your image based on the intensity or the distance. Okay, the kind of metric you use for deciding what to base your watershed on is independent of the watershed algorithm. The idea of the watershed is that you grow it out until you touch other cells and things like that. But the way you apply it can be different. And you'll see, if you look up watershed online, you'll see different examples and things like that. So I want you to know that the watershed is the growing bit rather than what you apply it to, which the most popular method is actually the distance transform. Okay, and you'll see the distance transform method in the practical. And if you want, you can actually change. You'll see I'm applying the watershed to the minus distance, and what you can actually do is you can apply it to the raw pixel data, and you'll see you'll get this as well. Both of them are watershed, but it's just how you apply it. Okay. So here's another example. So first we segment our image, we apply the distance transform. You can see it a bit better there. And then what we do is we find our seed points. That is basically just a threshold of the top levels of this. We take like the top 30% of it, and then we apply the watershed to this representation here, and we get our separated objects. And you'll see that in the practical. Okay. But this is a very common way of splitting objects when they're touching, based on their size. So if your objects are of relatively uniform size, then this will be very effective at separating them, even if they're touching. Okay. But this is classic kind of signal processing. It's well worth looking at these old algorithms and trying to understand them, because they're very elegant in many ways. Okay. So now onto some of these other segmentation methods. So a very common feature asked for in my institute is foci detection. And there was a particular PhD student that spent 2 years of his PhD counting foci manually in cells, until I came along and showed him the ImageJ plugin that does the same job in less than a second. And what is good about it is, despite some of these cells being brighter or darker than others, it still does a pretty good job at finding the foci. So it's, generally speaking, a good relative measure of intensity. And you can see basically you go from the raw intensities more or less straight to the foci, so there's no segmentation of the cells and things like that necessary for going and finding the point. So this is an interesting procedure to understand. And to understand it, we have to basically understand the building block of all foci detection algorithms, which is something called the maximum filter. So we've done some things about filters. If you're doing convolution, that is a linear filter. That's more or less the definition of a linear filter. This is a non-linear filter, which means it reacts differently depending on where it is. You can't go backwards with a non-linear filter. It's a one-way process. But don't worry so much about that today. What you really need to understand is basically a little bit about how it works. What we're going to do is we're going to take the small part of this image, which I represent like this, and we're going to apply the filter to this and see how it works. So imagine this little grid here from the image is just a 2D array of numbers. And what we're going to do is we've got a kernel of size 3, and we're starting in the corner. And the way that this maximum filter works, which is the beginning of the find maxima pipeline, is we're going to move this kernel around, and in the center of the kernel, in terms of our output image, so this output is actually relating to here, and this circle here represents our first output here. And what we do is basically we identify the maximum pixel within this neighborhood here, this 3 by 3 neighborhood, and we put it here in terms of our output. So whatever pixel is centered with our kernel, we look in the neighborhood and we assign that value, the maximum value, within the total neighborhood. Is that clear? Clear as mud? So I'll show you here as well so you can see. So we're going to move around this 3 by 3 neighborhood, and in the center, which corresponds over here to a different image, which is our output, we're going to put the value here, which corresponds to the maximum pixel in this neighborhood here. But it's kind of the center of the neighborhood, where it appears over here. So if we start moving through, you can see that here the maximum is actually shared between these 2 pixels, but that doesn't matter. We just assign in the middle whatever that value is, that is the maximum, and that goes in the output here. And then we keep moving. So here there are 3 pixels with the same value, but these are the maximum, and they go to this output here. And we keep moving round. Okay. And then here the maximum is this lighter grey here, so that goes there, and you can start to see that we're filling out our image. Here the maximum is here, so like that. And what we end up getting is from this input image, we get this pixel image here. Ignore the crosses just for the moment, but this is the maximum filtered representation of this image. So if you apply a maximum filter, which you can do very easily in SK image, scikit image, then depending on how big the kernel you use is, and we use the 3 by 3 kernel, if you put this image in with a 3 by 3 maximum filter, you'll get this image. Now that's fantastic, but what we want to do now is find the local maxima. And so there's a neat little trick that allows you to do that, and you have to think about this a bit maybe after the lecture, but why it works. But we take this input image and we compare this image to the maximum filter output image, and where the values are the same, those represent local maxima. Okay? And we have marked those with X's here. So where the pixel values are the same here compared to here, we mark them with an X, and those are our local maxima. Ta-da! Okay? So that's the beginning of our find maxima pipeline, and what we get is this. This is our local maxima. And you can see it's already starting to identify some of the foci, bright punctate points that we want in our cells, but there's also all of this stuff here as well. But these represent local maxima, but because the background's quite flat and a little bit noisy, we're also finding lots of peaks in the background. So although it works very well, and you can imagine in the 80s or 90s or whenever it was discovered, they were very excited by this. But what we realistically have to do in a practical setting is we have to prune these maxima in a sensible way just to get the ones sort of in the cells and the ones that aren't too close to one another, and we're not interested in the background. So a very simple way of doing this, and you'll see this in the practical, is just to take the foci beyond a certain intensity. You just say, if my foci, if this maxima is beyond a certain value, then we retain it. If not, then we get rid of it. But as I said, one of the strengths of this kind of approach is that you can find relative maxima even if the global intensity or the local intensity of the cell, say, is much lower than another cell. So we do this pruning approach, but there are many ways of doing it, and this schematic just represents that you can interrogate each of the peaks and their neighbours, and you can come up with a strategy which, with very few parameters, allows you to selectively get rid of the foci that you're not interested in, based really on whether they're within a noise tolerance of the other foci in that neighbourhood. So in the practical, as an optional exercise, there's a point where you can get another function which I wrote, which I copied from Fiji Image J and wrote in Python, which uses a very specific pruning strategy. And in the practical, if you want to do it, you can bring that into the Python notebook and apply it to an image and see how it works. Okay, but as it stands, this representation here is just the mean intensity plus 3 standard deviations as a means of just getting those foci which are really bright compared the rest of the image, which is a very simple way of doing it, which is quite effective. Yes?
Is there a difference in terms of efficiency, instead of doing these, to segment first with like a basic threshold and then find the maxima on the segmented image to avoid having to prune them?
Well, what's good about this is because you don't need to choose a global maxima, it's possible to separate the foci much more discreetly, because it's really just looking for that one pixel which is the peak. So you can do a global threshold, but then you're going to have to choose a value which kind of gets all of your foci, and you'll find quite a few of them just merged together. So this just allows you to be a bit more precise in what you're looking for. But yeah, that's a reasonable strategy, and it may in some cases be absolutely fine. But we found, especially for looking at DNA, probes that highlight DNA, that this approach was much more effective. And another kind of famous transform is the Hough line, well, the Hough transform, and here we've got the Hough line transform. And here what we do is we don't necessarily have to segment our image, but you can segment your image. But what we're looking for is a certain shape within our image. And what this does is it looks for straight lines within an image. Pretty simple. And what the intuition behind what the algorithm does is it just moves around the image and it tries to fit, it places a straight line basically in every pixel and rotates it. And if it gets a strong signal, in terms of it kind of correlates the pixels under the line, so if there's bright pixels there or thresholded pixels there, if it correlates with the position of the line, then it gives that a strong vote. And what you basically end up with is, once you've tried every possible line, direction and position in the image, you're left with the ones which vote very highly, and there you say that there is a line there. So you can see here, these green lines represents where it's found strongly voted for lines within the image. And you notice these lines are not continuous, there are gaps, but it doesn't matter because it's just put it across the image, and in a certain position there's enough pixels which line up in that direction for it to score highly as a line, and then you place a line there. You'll notice there are some lines which aren't so good, but this is because there are enough pixels moving across for it to get above the threshold. So that's a consequence of it, but you'll see in the practical later you can actually filter those out by looking at the angle of the line afterwards. So this is nice if your biology happens to form straight lines, but who's lucky enough for that to happen, huh? Only people that are streaking out DNA, it would seem. But if you have an opportunity to use it, it's lovely because you don't have to do. I mean, I did do segmentation on this, but it was a very simple segmentation, and in theory, you don't need to do a segmentation. You can do it on a grayscale image. So it's a very elegant thing if you've got the right biology for it. And then again, this is the the Hough Circular transform. You have to be very lucky to have this biology. This is GPMV, so these are vesicles derived from cells, and they are very, very spherical, and this transform works very well. And I came up with a nice kind of schematic for how this works. So in the previous case, what we did is we put lines everywhere and we moved them, we rotated them, and we found the top scoring location. This time we're going to pass circles of different size across our image, and where they fit very nicely with the grayscale representation, and that gets a high vote, and then we take them the highest voting locations for the circles of different sizes. And so look at it go, high voting locations. Now we're going to try a different size, see how they vote. Yeah, there's one there. And then we try the smaller circle. It's only through real passion do you get this good at keynote animations. And then once we've got the high scoring areas, we kind of choose a threshold and we retain those circles, and then we superimpose those circles over the image and can take measurements from there. And that, again, this is an optional exercise within the notebook where you can go and see how I've implemented it, but it's only if you have enough time with the other exercises. You might want to come back to it, but I've given you an image and a situation where it works. So now we're going to go away from these shape models to advanced numerical segmentation methods. So remember before I told you to think of your image more as a topographical representation. Level sets also borrows from this idea, but here we're going to say that our sort of brighter spots in our image are the tops of peaks. And what the level sets methodology does is it, rather than thinking of parameterized shapes, so in the previous examples we had circles and we had lines, but in reality a lot of the shapes we're looking at don't fit very well into that paradigm. So if you've got cells and they're touching each other, you would have to explicitly program a model that allows for that if they're touching and the different morphologies, and that's very complicated. So with level sets, we start thinking about things a little bit differently. We think about shape and form on a Cartesian grid, so we don't need to parameterize things, but we look at how these shapes evolve on this Cartesian grid, either with intensity or sometimes through time. There's a lot of ways of doing this depending on how you want to model your situation. And actually, one of the reasons we asked Virginie, who's coming tomorrow, is that she studied the mathematics of level sets and statistical shape models a lot. So she's going to go for a very elegant application of this kind of approach. But what I'm going to show you today is one algorithm in particular, which is have I got it here? This is actually from the Chan-Visa algorithm, which you'll see in the practical later. But what you tend to do, the kind of methodology of a level set method is we don't just look at the intensity of our object. We kind of define an energy term, and what we're trying to do is evolve this level set, which is parameterized on the Cartesian grid, on the pixel grid. We try and evolve it, taking into account different aspects of the image and of the level set itself. So we generally try and keep the level set to be of a certain smoothness that contributes to the overall energy. We want it to basically iterate until it reaches a place where the contrast, where the gradient is sharp in the image. So it's the edge of an edge of a cell rather than in the middle of the cell. Yes?
Is it the magic wand? Like on Photoshop
No.
It's not the same,
It's similar idea, but this is generally much more numerically intensive. But what that will basically do is try and match the intensities along, and maybe have a bit of smoothing involved in it as well. But this is a more numerically demanding, where we define this energy term, which can have a lot of different influences on it, like the smoothness of the contour, how the intensity changes if the texture is similar. There are many papers going into this and different approaches, and everyone thinks theirs is the best. But Virginie will talk more about that tomorrow. But it's sort of important to understand this is generally algorithms with many parameters, and there's ways of tailoring it to different images. And again, this is, at the moment, they're not as fashionable as they once were, because machine learning has come along and sort of transformed the field. but they're still very powerful. If you will need to encode very specific things about the shape of your cells and things like that, then you can do it through this methodology. Okay. So here is the Chan-Visa level set algorithm, which you'll see in the practical as an example. And what you do is you set parameters like lambda1 and lambda2, and I think I've explained them here. And there's certain ways of getting it to stop at a certain point. And if you're willing to invest the time in a particular algorithm, then you can get it to do amazing things. But because of the number of parameters and things like that, you have to invest some time to do it. But it can be very powerful. The thing is, machine learning has come along now, so people tend to learn from example rather than necessarily investing the time in learning exactly how these algorithms work. And I actually in that I'm in that case. But it will iterate until it gets to a certain level of accuracy on the image where it's no longer improving, and then it will stop. Okay, so I've included this in the practical on a working example, but Virginia will talk more about it tomorrow. Okay, so here is another advanced segmentation method. This is Superpixels, and this works very well on, for example, EM data, which we've got here. And this approach is a clustering approach, an unsupervised clustering approach. So we basically disperse cluster centers across our image, and then what we do is in each cluster center we try and recruit those pixels which are near to those cluster centers, but are also similar in terms of intensity. Okay, so it clusters spatially and in terms of intensity, and what this allows it to do is basically break up your image into nice what we call superpixels, and from here we can then make measurements in each chunk and potentially merge them together or do some kind of analysis on those individual chunks. And these are great fun to apply to photography images as well, and to biological images. And it's just a really nice way of breaking your image up into nice sized areas. It generally requires some processing subsequently to do things with those chunks, like merge them together. But it works surprisingly well at separating background from foreground, and also different parts of a cell from one another. Okay. And there are certain parameters, but not too many. So, like I said, you separate a number of clusters, distribute a number of clusters across the image. So you have to decide how many you're going to do that with. And then the process works better if it's smooth. So you define a sigma for the intensity of the strength of the Gaussian smoothing kernel. And then it has certain iterations in the clustering. And then compactness is a term which balances the weight between the importance of the color, the intensity, and the distance itself. Because these are two different notions, we have to weight them just like Stephen did with his tracking. And then spacing, it just assumes that your pixel grid is isotropic. So in microscopy, we generally do with anisotropic pixels because our Z sampling is generally worse than our XY, and this parameter allows you to balance those two forces as well. Okay, and then I'm not going to talk about it today. Mix is going to talk about it on Friday, but computer vision and machine learning are approaches which are really moving into segmentation in a big way. And like my poor knowledge of level sets shows, is that we're using these things more and more to solve segmentation tasks. And although level sets and statistical shape models still have a strong role, this has definitely been capturing people's interest in the last few years. And there are certain examples like UNet and StarDist and MassRCNN, which can be used for segmentation and are very powerful. But for these methods, you have to generally have a lot of training material, which can be annoying. With things like level sets and the other methods I've shown, you just need to tweak the parameters. You don't need explicit training material to use them. Okay, so they have that advantage. But as you say, we'll really focus more on this on Friday. There's a little animation. So then here is an overview of the methods. So there are many powerful methods for segmentation. There's no one answer to solving the question. The art is applying the right algorithm for the right problem. The more parameters an approach has, generally the more powerful the algorithm can be. There's no guarantees. And it's worth thinking carefully about which approach is best and when to use it. Two challenges. If you have enough data, most segmentation problems are solvable through deep learning and machine learning. But the challenge is it still requires a lot of annotated data, and people are trying to come up with methods that don't require as much annotated data. That's very much an active challenge. But for you moving into bioimage analysts, the challenge is really to be knowledgeable enough of the approaches that are there to apply them in a good way. So you notice when Stephen was talking about his segmentation, he used ISO data, was it? Which is a very kind of standard histogram interrogation, but it worked very well for what he did. So he used it because it's almost instantaneous processing, and it was completely sufficient. The worst thing you can do is throw the most advanced technique at a problem that doesn't need it. Thank you very much for your attention. Questions?